By the end of this session, you are supposed to have a brief understanding of the phases of analysis in a clinical laboratory, the possible errors that can occur in different phases of analysis, the test request form, sample rejection and the reasons for sample rejection, what quality control is, automation in clinical laboratories, and patient confidentiality. The pre-analytical phase starts when the clinician requests the test, which includes patient preparation, test selection with relevant clinical details, proper collection of the samples, and transport of the samples to the lab, sample reception in the laboratory, centrifugation of the samples if required, and sorting of the samples. Analytical phase includes sample processing till the final result is generated. And the final phase is the post-analytical phase from the result generation till the patient receives the report and the clinician interprets the report. Some of the possible common errors that can occur in the three different phases of analysis are suboptimal patient preparation, incomplete test request forms, using the wrong vacutainers, and improper transport of the samples that have been collected. The test request form should contain the necessary patient details along with the clinical details and the relevant history about the current condition of the patient. It should also contain the signature and name of the doctor requesting the test along with the date and time of request. Samples are accepted for analysis only if they meet a set of criteria. Some common reasons for sample rejection are incomplete test request forms, incorrect vacutainers used for blood collection, improper transport of the samples, missing test request forms or vacutainers, and insufficient quantity of sample. Quality control in the medical laboratory is a statistical process used to monitor and evaluate the analytical process that produces the patient results. This includes preventive maintenance, internal quality control, external quality control, and calibration. Automation in clinical laboratories over the years, with the advent of technology, has seen a shift from manual testing using colorimeter to a fully automated analyzer. An important aspect that needs to be kept in mind by us as doctors and laboratory personnel is the strict maintenance of patient confidentiality. This can be achieved by controlled access to patient data and dispatching the reports to the correct patient or treating doctor. The rest of the video aims to provide an understanding of the various phases of analysis in a clinical lab.
Good morning, madam.
Hello, sir. Please sit.
Thank you.
So, how have you been doing?
Things are all good.
Okay. So, you have come for the follow-up?
Yes. You had asked me to come after 3 months to follow up.
That's good. So, for your follow-up, we'll be doing two tests. One is fasting blood sugar, and another one is HbA1c test. This is to assess your current blood sugar and also the blood sugar status of past 3 months. For that, today you can have a normal diet, and after dinner tonight, you may not eat anything and come for your blood test tomorrow in the morning. You may have sips of water. So let me fill in your details.
At what time should I come in the morning tomorrow?
Uh, as early possible, you may come at 8 a.m.
Okay, so can I have tea, coffee, or something? Or
Uh, Better not have tea or coffee. You may have sips of water.
okay. It will be a blood test, no, madam?
Yes, your blood will be drawn for the test. So here is your request form.
Yeah.
You can take this for the blood test tomorrow morning.
Okay. All right. Thank you.
In the sample collection center, the phlebotomist checks the requested tests and generates a barcode which has the details required for the autoanalyzer to perform the analysis on the samples given. The same barcodes are stuck on the vacuitainers needed to collect blood for the relevant tests. For drawing of blood, the tourniquet is tied above the vein of interest to occlude the blood flow, the vein of interest commonly being the medial cubital vein. The area is then cleaned with rubbing alcohol and allowed to dry. Vein is visualized and pricked with a freshly opened sterile needle and the blood is collected in the relevant vacuitainers in the order of draw. Once the blood is drawn, the vacuitainers are gently mixed to ensure proper mixing of the additive, which is usually an anticoagulant, with the blood collected. Used needles are discarded in a blue puncture proof container. The blood samples and the test request form are then transported from the sample collection center to the clinical laboratory via a pneumatic chute system. Vacuitainers in their order of draw from left to right. A new sterile vacuitainer needle. A vacuitainer holder. Samples which need special care are transported to the lab in ice boxes. The samples arrive to the lab via the pneumatic chute. The box is opened and the samples and the test request form are taken out. The empty sample container is sent back to the sample collection center via the pneumatic chute. The barcode is scanned and the samples are received in the laboratory information system software. The received samples are then cross checked with the test request form to see if all the samples have arrived or not. The blood samples are centrifuged 30 minutes after sample collection at 5000 revolutions per minute for 5 minutes. If, serum and plasma are required, the vacuitainers are centrifuged. whereas for whole blood, the vacutainers are not centrifuged. After centrifugation, the samples are taken out of the centrifuge and the tubes are checked to see if the sediment or the clot is formed properly and the plasma and serum are clear or not. This ends the pre-analytical phase. Now coming to the analytical phase, the analysis of the sample is done using instruments like a colorimeter, which is based on the principle of Beer-Lambert law. The OD value or absorbance of the different solutions are measured using the colorimeter. The OD value of three different solutions are measured, being blank, standard, and test. The OD values measured of the blank, standard, and test solutions are noted down manually, and then they are used in a formula to calculate the concentration of the test solution. The semi-auto analyzer calculates the result or the concentration of the solution automatically by the formula that is pre-fed into the machine from the absorbance value that is measured by the machine. The different reagents are then manually added using the auto pipettes into the test tube, mixed thoroughly, and then the sample is added. The sample reagent mixture is mixed. Where the volumes that has to be aspirated by the pipette are already preset. The reagent and sample mixture is mixed properly. The sample reagent mixture is then aspirated into the semi-auto analyzer, which measures the absorbance value and calculates the result. The measured absorbance value along with the calculated result are displayed by the semi-auto analyzer on the screen. The C is the concentration of the sample. In a fully automated auto analyzer, the samples are placed in the sample racks of the analyzer. They are placed in such a way that the barcode is clearly visible in the slit. And then the racks are placed into the auto analyzer and the machine is started. The machine automatically takes in the samples and it scans the barcode of each sample, identifies what tests have been requested for that particular sample, and starts processing them. The reagents required for processing the samples are already placed inside the analyzer. It will pick up the required volumes of the reagent and sample and add it to a measuring cell. A reaction is performed and the result is generated automatically by the autoanalyzer. Various quality control measures are in place to ensure the accuracy and precision of the results generated by the autoanalyzer. These quality control measures are such as the internal quality control, external quality control, and frequent calibration as and when required. The autoanalyzer generates the result after doing the calculation, and these results are displayed on the data manager, which are verified by the technician.
This is the complete certified report. As you can see, the patient details are shown here. A unique lab number is shown here. The date of collection as well as time of collection is shown here. Sample reception time and date is here, and from which ward testing is also here. You can see 4 different columns here: the test, for the result, for the reference interval, and the method used. Now all the values are given here. If there is an abnormal value, it would be highlighted. If it is red, it is high. If it is blue, it is low.
Coming to the post-analytical phase, this area is where the authorized signatories check the reports. They check the reports in the laboratory information system, and then they certify the reports, which can be dispatched to the patient.